Good day. Thank you for tuning into this presentation. My name is Anita Moretti, a sophomore at the Adventist University of the Philippines, studying medical laboratory science. I will be presenting a summary of a critic paper that I prepared related to recurrent fish abnormalities and myeloid disorders. The title of the journal article I found in relation to that is Cytogenetic Profiling in Pediatric Acute Leukemia, a report on 746 newly diagnosed pediatric cases analyzing the spectrum of recurring chromosomal rearrangements in B-cell lymphoblastic and acute myeloid leukemia, leading us into the introductory part of the critic paper. And so in the journal article, there were three main keywords that were used. The first one is acute lymphoblastic leukemia, which is a type of cancer in which the bone marrow makes too many lymphocytes according to the National Cancer Institute. The second keyword is acute myeloid leukemia, which based on research is defined as a malignancy, basically just the presence of cancerous cells of the stem cell precursors of the myeloid lineage, such as the red blood cells, the platelets, the white blood cells, other than the B and the T cells. Then lastly, we have cytogenetics, which involves genetics, inheritance, or the study of chromosomes, and its relation to this is FISH, which stands for fluorescence in situ hybridization, and this is basically just a cytogenetic technique that detects specific genes within a chromosome and a DNA sample, as well as any irregularities that might be there in the DNA. And also in the study, karyotyping was also used, which examines the cells in a chromosome. So this one here is just a demonstration of the steps in FISH, and this one is just to give us an idea of what a karyotype looks like. Still on the introduction, the study aimed to assess the prevalence of different cytogenetic abnormalities in pediatric acute leukemia in Pakistan and compare it to global trends. And so it investigated the occurrence and the comparison of different cytogenetic abnormalities in pediatric acute leukemia, specifically B-cell acute lymphoblastic leukemia, also known as BL, and acute myeloid leukemia, known as AML, just abbreviations to these, as I already defined them, and to help us understand these myeloid disorders further. And according to the study, cytogenetic advancements in acute leukemia have provided valuable insights into molecular mechanisms that play a crucial role in the diagnosis and prognosis of both the pediatric adult acute lymphoid and the acute myeloid leukemia AML. Thus, through this critic paper, we will weigh the strengths and the weaknesses of this study. As for the methods, the study was conducted at Indus Hospital and used a total of 746 patients who were diagnosed with acute leukemia between January 2015 and October 2018. FISH and karyotyping were also utilized, and patients under the age of 18 years were used for the study. So in order to analyze the data, a statistical software called SPSS version 2.0 was used for this one. Of course, there are limitations to the methods that are used in the study, such as like selection bias, as the study included patients from a specific hospital or region, leading to potential selection bias, thus limiting the applicability of the results to a broader population. Also, it limited the ability to establish causal relationships between cytogenetic abnormalities and outcomes in pediatric acute leukemia. Results and discussion. There were many findings in the study. Of the 746 patients, 531 were diagnosed with B-cell acute lymphoblastic leukemia BL and 215 with acute myeloid leukemia AML. And among the BL patients, there were 321 males and 210 females, with a male to female ratio of 1.5:1, implying that males had a higher result than the females. The other findings were that in AML, 70 patients had recurrent cytogenetic abnormalities, and abnormalities were detected in 7.9% of AML cases. And one interesting thing too is that interface FISH analysis also revealed detectable recurrent genetic abnormalities in 32% of tested BL patients. Due to the karyotyping analysis results, the study conducted a comprehensive cytogenetic analysis of pediatric leukemic patients to understand the association of various factors such as age, gender, and genetic factors. The study also found that the prevalence of certain chromosomal abnormalities in pediatric AML, which is acute myeloid disorder, was comparable to Western and Asian populations. Thus, when we recall one of the aims of the study, we can see that this study was compared to other areas. But then, even though the study aimed to improve understanding of the biology of the disease to enhance care and management of better survival outcomes, the challenge could have been that there wasn't enough local data. This is a possible limitation to the study. Conclusions, recommendations, and future directions. In conclusion, there was a high prevalence of cytogenetic abnormalities in Pakistan, and there was more to the findings since different genetic makeups have a role to play in the underlying genetic susceptibility, and gender and age played a role in the findings of the study. The lack of comparable results for FISH and karyotype, or the absence of karyotype testing in cases of acute myeloid leukemia and T-cell acute lymphoblastic leukemias, limits one of the objectives, which was to investigate the occurrence and comparison of different cytogenetic abnormalities in pediatric acute leukemia, more specifically B-cell acute lymphoblastic leukemia, BL, and acute myeloid leukemia, AML. My recommendation or suggestion, however, is that all factors that affect prognosis of the disease should be considered, and the study should not be focused on one area, but globally. But why am I saying this? In order to get accurate results in assessing genetic makeup, the environment, diet, psychological state, gender, age, and so on in relation to the recurrence of leukemia. This is just the list of the references that I used in this critic paper. Thank you for listening till the end.